Welcome, Life Sciences, to yet another wonderful life science lesson. We are on the journey of the microorganism. In this series of lessons, we're looking at the concepts of what microorganisms are and especially what they can cause. And we're looking specifically at diseases and ones that are found in South Africa particularly. If you had followed the last lesson, you would have looked at viruses, right? Very, very, very important microorganisms. I'm sure you've heard of COVID. If you've lived through COVID, you understand what the concept of a virus is. Today, we're going to look at the microorganism that we call bacteria. Now, if you have a look on the board here, I've posed a question, and I've said to them over here, that they can make us sick, right? But we cannot do without them at all, right? We need bacteria in our lives. Although we're going to look at the ones that are going to be pathogenic, bad for us, right? Bacteria are essential, right, for us to actually live healthily. But unfortunately, there are some that we're going to look at that are going to make us sick. All right, so let's take a look at what bacteria are. Oh, we got to there we go. So if we have a look at our concept map, what are we going to look in today's lesson? It's very similar to the concept that we look at when we looked at viruses. So we want to look at a bacteria. What must you know? You must know what they look like. You must know their characteristics. How do we mean by that? Is how they operate. How do they function? Maybe knowing that, what can we do? Can we cure them? Can we kill them? Can we get rid of them? And in that vein, I'm going to go over 4 diseases with you. But you just need to know one of them. You don't need to know all 4 of them. And when we look at diseases, you need to know what effect does that disease have on me and how am I going to manage the spread of the disease. Okay, so let's start looking at this concept of what bacteria are. Okay, I've got keywords on the board here. I'm not going to go through all of them individually. As you know, right, as we go through them, right, we're going to discuss those terms. Okay, so when we look at bacteria, maybe in previous models, you might have looked at the section called the classification and biodiversity. Now, classification is all living things on this planet are put into different kingdoms. And basically what it is, it's a checklist. So if you've got this, you go into this kingdom. If you have this criteria, you go into that kingdom. And you might have done, all right, the kingdoms that you would have looked at was plants, animals, fungi, protists, right? And what we're looking at today, Monera. The kingdom Monera is where bacteria fits in. And those specific characteristics mean that all bacteria have got certain things, characteristics that put them into that particular kingdom. Just have a look at a few that I'm hoping might ring a bell. Okay, so guys, if you belong to the kingdom Monera, and let me get my pen ready here, choose my beautiful blue. Right, we've said, there we go, the kingdom Monera. There are a few words that I just want you to, right, be able to remember. They are unicellular. Unicellular means one. You're going to see now when we look at the structure, bacteria are only one cell. They are also the only cells that are prokaryote. And I don't know if you remember it. What is a prokaryote cell? It's a single cell that has DNA in the middle, and it has no nuclear-bound structures. So it will have a ribosome, but it won't have anything else. There won't be any Golgi body, there won't be any nuclear membrane, there won't be any mitochondria. Okay, so that's where we're looking, prokaryotic. When we come to bacteria, we said most are useful, But remember this term pathogenic. What does pathogenic mean? They're going to produce disease. Now, the very last point is really important when it comes to this. We're not going to look at it during this lesson, but we might look at it, we're going to look at it in further lessons. Guys, bacteria can be killed by antibiotics. When we looked at the lesson in this model before this, when we looked at viruses, viruses couldn't be killed. Right, so we can use vaccines, etc., to try and build antibodies, but a bacteria is a living cell, and we have medicines, antibiotics, that can kill them. Okay, so let's have a look at what a bacteria cell looks like. A bacterium, singular, and we're going to have a look at a typical cell. Okay, so guys, let's have a look here. So when it comes to the structure, as I said, it's prokaryotic, and what it has, it's got 3 layers around it. Can you see here? It's a cell membrane or a plasma membrane. So this is the one layer. All living things have a cell membrane. Then it has a cell wall around it. It's not a plant cell. Yes, plant cells have a cell wall, but this is not a plant. It doesn't have characteristics of plants. And then you'll see a very special thing around the 3rd layer is a capsule. And basically what that means is sometimes a bacteria cell can actually form like a spore. That capsule can form a hard cover, and the bacteria can be dormant. Dormant means it's not doing anything, and it can just lie there until conditions are ideal. And when conditions are ideal, that bacteria might emerge from its capsule. Okay, what else? Very important, in the middle, the cell, we have the DNA. Unlike a virus, which is DNA or RNA, it's the genetic material. All right, and that is important. Living things need to reproduce. Now, we've got, if you look inside here, we have got ribosomes, and ribosomes are organelles, but they do not have, all right, a membrane, and that is why they are in, right, the bacteria cells, and they are there because they are going to make protein, very important. Now, when we have an antibiotic, the antibiotic stops these ribosomes from working. If it can't make a protein, you're going to see it's going to die. Now, this is a little tail on the end of a bacterium. Not all bacteria have tails. Okay, a tail is there if it needs to swim, and not all bacteria need to be able to swim. Okay, so guys, you really need to know the structure of a bacterium. You might be able to, well, you might have to draw it, but you definitely need to know the labels and the functions for each of the different parts. Okay, the same as you had to do for when you do the virus. Now, when we look at bacteria, you will see they are actually classified according to their shapes. And we usually find 3, sometimes 4 different shapes of bacteria. And we often give them the name of the cell, right, according to the shape of the different bacteria. Let's have a look at the words. Okay, so if, over here, if they are rod-shaped, can you see these are like that? Then we call it a bacillus. And you're going to see a lot of the bacteria that we're going to look at today are bacillus cells. If they are round, they are called cocci. All right, and lots of you, right, have actually had this before. And it's a streptococcus. And you're all thinking, oh my soul, what is that? It's very simply a sore throat. Okay? You can get a sore throat, and that bacteria that attacks your throat and makes it sore is called a streptococcus. And I don't know if you might have a strepsils if you have a sore throat. That will help you with that. And the very last one over here is a spirille, a spiral. Okay, and this one over here, unfortunately, that is an STD, and that is syphilis. All right, syphilis is a spiral, and it's a sexually transmitted disease, and these are all bacteria. And as I say to you, all the bacteria that we are looking at, you can treat with, right, antibiotics. So that is going to be a good sign. Okay, now, bacteria are living, which means they can reproduce. Unlike a virus, when we looked at last time, they had to actually go to a host cell to be able to reproduce. Bacteria reproduce on their own, and they do it very, very quickly. And that's why we can use a lot of biotechnology. We can use bacteria to actually make things that we need. And we'll look at that in later lessons when we look at how we can use bacteria to make human insulin. So bacteria don't need a host, right, like the virus did. Basically what they're going to do, it's a process called binary fission. So you need to know the term, okay? That's what it's called. We don't call it mitosis, and we don't call it cloning, although that is actually what it is. Okay, so very simply, if you'll have a look here, all that a bacteria cell is going to do, here's the cell. It's going to make a copy of its DNA so that it can put one copy in one side, one copy in the other. The cytoplasm is going to divide, and what do we have? Probably two identical bacterium cells. So nice and easy, right? And that is why you will notice if your bacteria, once it starts to grow, right, grows very, very, very quickly, especially if it's a nice, warm environment. That's why we put our food in the fridge, right, so that we can stop it or slow down the process of it dividing to make more copies of itself so our food doesn't go off. Okay, guys, that's it for now. We're going to take a quick little break, and we'll see you right back just now.